Your immune system is arguably the most sophisticated security apparatus in the known universe. It operates continuously, distinguishing between your own cells, harmless foreign substances, and genuine threats, all without conscious instruction. Every second of every day, your immune system is scanning, responding, remembering, and adapting. It is not a single organ but a distributed network spanning your bone marrow, thymus, spleen, lymph nodes, tonsils, gut lining, and the bloodstream itself. The cells that carry out immune functions are all derived from stem cells in the bone marrow and collectively number in the trillions. Understanding how this system works mechanistically, not just the vocabulary but the actual biology, reveals why vaccines work, why we get sick, why autoimmune diseases happen, and why cancer can evade detection. Immunologists divide the immune system into two broad arms, the innate immune system and the adaptive immune system. The innate system is your first line of defense, fast, nonspecific, and present at birth. It responds within minutes to hours by recognizing general molecular patterns that mark things as foreign. These patterns, called PAMPs, pathogen-associated molecular patterns, are detected by receptors called toll-like receptors on immune cells like macrophages and neutrophils. When a toll-like receptor detects a PAMP, it triggers a cascade of inflammatory signals. Neutrophils, the most abundant white blood cell, rush to the site of infection and engulf or destroy pathogens. Natural killer cells patrol the body looking for cells that have lost a surface marker called MHC class I, a telltale sign that a cell has been infected by a virus or has become cancerous. The adaptive immune system is slower, taking days to weeks to fully activate, but it has two capabilities the innate system lacks: specificity and memory. The adaptive system's main players are lymphocytes, B cells and T cells. B cells are produced and mature in the bone marrow. Each B cell carries a unique receptor on its surface that can recognize one specific molecular shape, called an antigen. When a B cell encounters its matching antigen and receives the right co-stimulatory signals, it differentiates into a plasma cell that secretes antibodies, Y-shaped proteins that circulate in the blood and bind to that antigen with high specificity. Different classes of antibodies, IgG, IgM, IgA, serve different functions. IgM is produced first in an immune response, IgG is the most abundant and provides long-term immunity, and IgA is concentrated in mucosal secretions like saliva and breast milk. T cells mature in the thymus, the organ that gives them their name. There are two main functional types. CD4-positive T cells, called helper T cells, do not directly kill pathogens but orchestrate the immune response by releasing signaling molecules called cytokines that activate B cells, macrophages, and other immune cells. CD8-positive T cells, called cytotoxic T cells, are the killers. They recognize infected or cancerous cells by detecting fragments of foreign protein presented on MHC class I molecules on the cell surface, and then deliver a lethal package of proteins that punctures the target cell's membrane. MHC molecules, the major histocompatibility complex, function like molecular display windows, showing neighboring immune cell samples of whatever proteins the cell is currently making. If a virus has infected a cell, viral peptides appear in those windows, flagging the cell for destruction. Fever is one of the most misunderstood immune responses. When the innate system detects infection, immune cells release signaling proteins called pyrogens, including interleukin-1, interleukin-6, and tumor necrosis factor alpha. These reach the hypothalamus in the brain, which acts as the body's thermostat, and raise its set point. The result is a fever. Your body shivers to generate heat and constricts blood vessels to conserve it. Far from being simply a side effect of illness, fever actively inhibits the replication of many pathogens, which are adapted to function at normal body temperature, and enhances the activity of immune cells. The memory that makes second exposures to a pathogen much milder is stored in long-lived memory B and T cells that persist for years or decades after the initial infection, primed to mount a faster and more powerful response if the same antigen appears again. Autoimmunity occurs when the immune system loses its ability to distinguish self from non-self and begins attacking the body's own tissues. This is not a malfunction from outside, it is a breakdown in the system's own training. During T-cell development in the thymus, T-cells that react too strongly to the body's own proteins are normally eliminated in a process called central tolerance. When this process fails, self-reactive T-cells escape into circulation. In type 1 diabetes, autoreactive T-cells destroy the insulin-producing beta cells of the pancreas. In rheumatoid arthritis, the synovial membranes of joints are attacked. In multiple sclerosis, it is the myelin sheath around neurons. Understanding the immune system's normal logic, including how it decides what to attack and what to spare, is the foundation for understanding not just autoimmunity, but allergy, transplant rejection, cancer immunotherapy, and nearly every modern advance in medicine.